Hello students, today's topic is regulation of gene expression. It is multi-step process by which a gene is regulated and its product is synthesized. Thus gene expression results in the formation of a polypeptide. Genes of a cell are expressed to perform different functions. For example, an enzyme beta galactosidase is synthesized by E. coli is used for hydrolysis of lactose into glucose and galactose. Certain bacteria like E. coli adapt to their chemical environment by synthesizing certain enzymes depending upon the substrate present. Such adaptive enzyme is called inducible enzyme. A set of genes will be switched on when there is necessity to metabolize a new substrate. This phenomenon is called induction, and small molecule responsible for this is known as inducer. It is positive control. Let's watch the given image of regulation of gene expression by a hormone receptor. Then watch the given video to understand the concept of regulation.
Understand about a very important topic, the gene regulation and lack of it. So without further ado, let's first start with gene regulation. Gene regulation, in the most simplest terms, is the ability of an organism to control or regulate what proteins it makes from its DNA. In our body, we have numerous types of tissues and cells. We have muscle cells known as myocytes, we have cells in our intestines known as enterocytes, and the cells in our skin known as keratinocytes. All these cells are different in shapes, sizes, and functions as well. The myocytes' main job is muscle contraction, enterocytes produce enzymes for food digestion, and keratinocytes form a barrier known as skin. All these cells contain the same DNA in them. However, they are different in sizes, structure, and function due to the different proteins they produce. As most of you may know, our DNA is made up of thousands of nucleotide bases which are paired, and these are the basic units of DNA. The human DNA contains over 3.1 billion such base pairs. These base pairs are arranged in groups known as genes. Now, if you take out the DNA from the nucleus of a muscle cell, a skin cell, and a cell from the intestine, they would contain the same 3.1 billion base pairs of nucleotides arranged in the same sequence. But despite having the same DNA in them, they are completely different, and this is because of the different proteins they produce. This is all possible due to the ability of the cell to control or regulate what proteins it makes from its DNA. In a broad sense, the genes in our DNA can be divided into two main groups: structural genes, which code for proteins, and regulatory genes, which regulate the proteins that are produced by the structural genes. We have about 20,400 structural genes in our DNA that code for the various proteins. But you would be surprised to know that makes up only about 2% of our DNA. The vast 98% of our DNA does not code for any proteins and is mostly regulatory. This non-coding regulatory DNA is responsible for gene regulation. Through this process of gene regulation, a cell can also increase or decrease the production of a specific protein when it needs. For example, the B cells in your body produce antibodies that fight off infections. The antibodies are also proteins and are produced at a relatively low level in the absence of a particular infection. But in the case of an infection, the production of antibodies can increase many times over, all through the upregulation of genes that are producing these antibodies. Another common example includes the beta cells in your pancreas that produce the hormone insulin. Now, these cells do not produce insulin at a constant rate, but rather increase or decrease the production of insulin depending upon the amount of glucose that is present in our bloodstream. This is possible because the beta cells of pancreas have special receptors in them that enables them to sense the amount of glucose in the blood. When the blood glucose rises, it is noticed by these receptors which sends the information to the cell's DNA. The regulatory genes located behind the insulin genes note this and increase the amount of insulin that is being produced by the structural genes. This is one simple example of gene regulation. Gene regulation works in thousands of ways, but in this video we will look at a very basic model of gene regulation that operates in a bacteria known as E. coli, and this is known as the lac operon. Now there are many things that I would like to explain before beginning with the lac operon, but one important concept here is the central dogma of gene expression. Now to keep it very simple, the central dogma basically says that the information is copied from DNA to RNA, which is known as transcription, and from RNA to proteins, which is known as translation. So the gene regulation can work at multiple levels in this model of gene expression, but the most common point where the gene regulation operates is at the level of transcription, the point where the information on the DNA is copied onto RNA. Now, like we discussed earlier in this video, we have structural genes that code for proteins, and then we have regulatory DNA which regulates the proteins the structural DNA makes. Let's look at how this regulatory portion of DNA is usually arranged and what is the most common mechanism in gene regulation. Now, suppose we have two genes on this chromosome that code for protein A and protein B. Just before these two protein coding genes, we have the regulatory DNA that is shown in blue, which controls these genes. The regulatory portion of DNA is usually arranged into a regulatory sequence that is shown in light blue and regulatory gene which is shown in dark blue. The regulatory sequence is usually a few hundred to thousand base pair long sequence that lies just behind the main protein coding genes, and it is the site of attachment of the enzyme RNA polymerase. This enzyme copies the information on the genes A and B onto an mRNA, that is the process of transcription. The main regulatory gene usually lies a few thousand base pairs behind the protein coding genes, and this regulatory gene synthesizes a protein known as the regulatory protein. The regulatory protein goes on and controls the binding of enzyme RNA polymerase to the regulatory sequence, increasing it or decreasing it depending upon the situation. So this is a basic model of how gene regulation works at the level of transcription. We have regulatory genes that control the rate of transcription of the protein coding genes, also known as the structural genes.